Good afternoon, everyone, and welcome to today's local government education program provided by University of Illinois Extension. My name is Lisa Merrifield, and I'm a Community and Economic Development Specialist with the University of Illinois Extension. For sound quality, please mute your microphones during the presentation. I also need to mention that during this time of increased Zoom use, we are occasionally having people join meetings with malicious intent. So please note that the moderators will be monitoring the chat box throughout the presentation, and we will remove anyone who posts any inappropriate content as quickly as possible. We will also not be posting any links in the chat box today. We will email anything to you after the presentation. But if you see a link, do not click on it. It is not our intent to post any links in that chat box. If you have any questions, though, please use the chat box. I will be monitoring that, and I will ask Holly any questions that you have after the end of the presentation. All right, a recording of the webinar will be made available on our local government education website and our YouTube channel following the presentation. Participants will also receive slides, links to resources, and a recording for today's webinar. Today, we will hear about tick management from Dr. Holly Tutton. Holly is a vector ecologist with a natural history survey. She conducts research on ticks and mosquitoes and runs the Illinois statewide active tick surveillance program through a contract with the State of Illinois Department of Public Health. Holly, at this time, I'll turn it over to you.
All right, thanks a lot, Lisa. Lisa, can you hear me, just to be sure?
Yep, we can hear you.
All right. So I just want to say good afternoon and thanks to all of you for attending the webinar. I'm going to spend about 40 minutes talking about ticks and tick-borne diseases in Illinois, our surveillance efforts, and what you can do to prevent tick bites. We'll have time at the end for questions. I've scheduled myself out beyond the 1 p.m. period so I can answer any questions there are. And also in the future, if you have questions or comments, please feel free to reach out via email to contact me. I just want to say I'm really thrilled to be here sharing this with you today, and I'm glad that we connect like this online. So today, I'll generally be talking about 4 broad topics, and I'm going to be providing a lot of links to resources that you can use to extend what I'm presenting. You'll get a copy of this after the webinar, and you'll be able to access those links. So some slides might look information dense, but you'll have time to review them later. I'm going to introduce our active tick surveillance in Illinois, highlight some species and disease agents to watch out for, present information on ticks, climate change, and landscape management. And then I'm going to end with a quick section on tick bite avoidance and proper response methods to tick bites. And then, like I said, if you have any follow-up questions, please feel free to email me. So to start off with ticks and tick-borne disease agents in Illinois, we have 5 species of greatest concern and some emerging diseases that I think it's worth knowing about. The structure of our active tick surveillance program is that it's the first statewide tick and disease agent surveillance program for Illinois for ticks. It began in mid-2019. And it consists of people from our lab or stakeholders at local departments of public health or forest preserve districts going out into the environment and actively looking for ticks. The CDC has a defined protocol for this. We collect the ticks that we find in the environment, and then they get sent back to my lab where I identify them and we test them for different disease agents. Those tick species that we find and the disease agents are posted online. And I'm going to take you through the website where we have these maps. I think that this is an outstanding resource that's being provided by IDPH. It's data from our collections and our tick testing that we are sending to our partners at IDPH, and then they're doing the hard work in the background to get it up online for the public to use. However, there are some nuances to the website, so I'm going to run you through a video so you can see how you can access those data. So you'll see my cursor in the middle of the screen there. I'm going to turn it on. It's going to start moving in a second. Like I said, this is at IDPH. It's hosted by IDPH, by the excellent vector control specialist there. It's a public website anyone can access. I'm going to show you how to navigate to it in a minute. The data are regularly updated from our tick collections and tick testing. And you can get information about different species of ticks by clicking on the tabs at the top there. You can also get information about the tick itself, its biology, the different pathogens and disease agents we're finding in these ticks. And it's aggregated at a county level. So if you have a site that's participating, your individual site won't be implicated. You can see what the status of the tick species is in a given county based on the color, and you can get more information about the biology, behavior, associated disease agents of that particular tick species when it's active, and links out to more information from CDC. So if we look at a particular disease agent, such as the agent of Lyme, Borrelia Miyamotoi, you can see that by clicking on the tab, you can get more information about that disease agent. And by scrolling down, you can learn some very valuable information like the incubation period, signs and symptoms, and even information on treatment. So this is also a great resource for medical professionals. You'll get links out to IDPH, to CDC, and also a link to our lab if you need to contact us. If you click on an individual county, then you can see for that particular disease agent how many ticks we tested and how many were positive for it. And like I said, this information is regularly updated. We just started the program, so those will increase. You can also look at tick habitats. This is a fantastic resource. You're going to see it in just a second. When you zoom into the map, you can look at different land cover designations. So is it forested, is it agricultural, and you can estimate the risk associated with them in your county. So you've got to zoom in. You always have to zoom in when you get to this tab. But then you can see that there are forested areas, they're considered high risk, and in Cass County, this is where the forested areas are. You can even zoom in at a much finer scale on this map. I'm just showing it to you right now. Again, this is an outstanding resource being provided by IDPH, and I encourage everybody to access it and tell your friends about it. Now, it's not too easy to find, so I'm going to show you a very quick and simple way to always be able to get to this map. You'll see the cursor move. You go to dph.illinois.gov, that's the IDPH main website. Topics and services, there's a drop-down menu. Go to environmental health protection. Go all the way down that alphabetical list to vector control and surveillance. Click on it. And then go all the way over to the right to the Illinois tick surveillance map. Click on that. And wait a couple seconds for the data to load. And you'll get to that map that I just ran you through. You can also find it. I dropped the actual link to it in this presentation, which you'll get a copy of the presentation later. You can enter that into your web browser, or you can go to a web search engine and type in Illinois tick surveillance maps. Now, sometimes it's the top hit, sometimes it's the 7th hit. It just depends. So moving on to information about the ticks themselves. The ticks that we're most concerned about in Illinois in terms of human or even companion animal problems are 3-host heart ticks. That's what the group is known as. They're very efficient vectors because they go through 4 life stages: egg, larva, nymph, adult, and require a blood meal from a new host to advance to each new stage. So larvae require a blood meal before molting to nymphs. Nymphs require one before molting to adults, and adult females require massive blood meal to gestate eggs. Most adult males will also take blood meals. So this accumulation of hosts over a tick's lifetime also means that they can accumulate disease agents as they grow. Humans are typically bit by nymphs and adults, and that means ticks have already taken 1 to 2 blood meals before biting us. So for some specific species and disease agents of concern, I'm going to start with the Lone Star tick, Amblyomma americanum. It appears to be most abundant in southern Illinois, but it will likely spread north across the state and eventually be abundant throughout. It's also known as the turkey tick or turkey mite, and larvae can sometimes be confused as chiggers. They're very aggressive biters, and all life stages will bite humans. You can see down there a small box on the seasonality of the different life stages. They are associated with the disease agents of human ehrlichiosis and tularemia, and two new emerging diseases that I'll talk a little bit more about right now. So if you look over on the left, you'll see a map, and that's where we found ticks that were positive for Heartland virus in Illinois last year. Heartland virus is an emerging disease in Illinois and in the US in general. It was only recently discovered in 2 patients in Missouri. Extensive field surveys implicated the Lone Star tick as the vector, and Illinois had 2 human cases in 2018. In 2019, we conducted tick collections at the suspected sites of tick bites, and the ticks we recovered from Kankakee and Williamson counties, very, very far apart across the state, were positive for this virus. So we have a publication on the investigation that will be in print soon, and if you want a copy, just email me. And then moving on to a second disease that's associated with Lone Star ticks, the alpha-gal allergy. This one is a really, really weird one. It presents as an allergy to mammal meat and mammal byproducts. The symptoms can be as severe as anaphylaxis, or they can present as ongoing vague gastrointestinal distress. So essentially, it's associated with the bite of the Lone Star tick in the United States. A few weeks after that bite, you might eat a burger, and then a few hours after that burger, you might not feel so good. And it could indicate that you have this new allergy or sensitivity to mammal meat. There is a definitive test for humans available. Allergists tend to be the most well-informed on them, so if you are interested in more information, reach out to an allergist. And data recently shared at CDC Vector Week that was based on human test results indicates that this disease occurs in Illinois at a higher percentile than in other areas of the country. So it's something to be thinking about. So moving on to the next species, the Gulf Coast tick, Amblyomma maculatum, was recently shown to be established in Illinois, and I'll link to that publication in the next slide. Very little is currently known about its distribution, but we are actively planning surveillance for this species and think it might be focally distributed in the States, not all over the place, but potentially in very specific areas. Adults are the life stage that tend to bite humans, and they are fairly aggressive biters when encountered. Unlike other tick species in the state, which are usually associated with forests, this species can be found in sunny, dry areas, such as prairies or roadsides. Its overall appearance is quite similar to the more common American dog tick, and that nuance might be overlooked even by someone familiar with tick identification. If you do think you've encountered the Gulf Coast tick, please send me samples or contact me for more information. So a disease agent that's associated with the Gulf Coast tick, Rickettsia Parker Eye, is an emerging pathogen in the United States that's transmitted by that tick. We recently tested Gulf Coast ticks from Southern Illinois and found a very high incidence of this disease agent in them. The publication is printed and available in the Journal of Parasitology or by contacting me. We also have a blog post at the University of Illinois. If you look to the very bottom of the slide, there's a link to that blog post as well that you can share and read. So this disease agent in humans causes symptoms very similar to Rocky Mountain spotted fever, often described as milder though, and often presents with a scab at the bite site called an inoculation eschar. Very importantly, no specific test for this pathogen exists, but it can cause positive results on a test for Rocky Mountain spotted fever. Thus, some of our human cases of RMSF in Illinois might actually be the rickettsiosis that's caused by this disease agent. So moving on to the tick that most people are pretty familiar with, the American dog tick. It's abundant and common throughout the state. The adults tend to be the life stage that bites humans, and it is the vector of the bacterium that causes Rocky Mountain spotted fever, a very, very serious illness that can kill people. This tick has a very wide range of habitats and readily bites people. And finally, well, this is the fourth 5. The black-legged tick, it's the vector of several human disease agents that often cause debilitating illnesses, including Lyme disease. And it can transmit more than one disease agent in a single bite. During the course of our surveillance so far, we have significantly expanded knowledge of where this species and the disease agents it transmits occur in Illinois. You can see that on the map that we're noting it much more than just the north, where it was previously considered to be established. For this species and others, I do encourage you to check out that interactive map at the IDPH website. Some points that are worth noting. Adults of the black-legged tick can be active over the entire winter on any day with little to no snow or rain when temperatures are above around 40 Fahrenheit. Keep in mind, this can be when the temperature becomes elevated in very distinct microclimates. I've collected adults several days after heavy snowfall on southern-facing slopes with strong sun. Nymphs are considered the life stage of highest disease transmission risk because they are easier to overlook during a tick check. So we might have another emerging tick-borne pathogen in Illinois, Borrelia Miyamotoi. It's in the same group as the bacterium that causes Lyme. It's transmitted to humans by the same vector, black-legged ticks, and we have found it in ticks in Illinois. It does not cause Lyme, but the symptoms of the disease it causes, such as fever, chills, and joint and muscle aches, are very similar to Lyme. However, it is not associated with a rash like Lyme and will not typically cause a positive result on a Lyme test. This presents us with a challenging diagnostic scenario. A person might have a documented black-legged tick bite, have many symptoms of Lyme but no rash, and have a negative Lyme test, but still have an active tick-borne infection. A human test does exist for this disease agent, but it has to be ordered by a doctor. And then finally, the last tick I want to highlight in today's talk is a new invasive in the United States, the Asian longhorned tick, Haemaphysalis longicornis. Now, it's not in Illinois yet. It's likely only a matter of time before it shows up here. It was recently introduced to the United States and has so far been found on a wide array of hosts, including humans. It has not been associated with any human diseases in the US, but once it reaches an area, it has a great potential to spread because the females can reproduce without needing a mate. This tick could be confused with other species, so if you think you have encountered one, please consult an identification expert. Which brings me to my wrap-up of this section. I do want to point out that at our lab website, we have instructions for sending in loose ticks, like a tick crawling in your pant legs or in your pet's fur. If you have found a loose tick and would like an identification of it, feel free to send it to me. And I've got the link to our website at the top there. So moving on to our next section, ticks and climate change in Illinois. You know, regarding ticks and climate change in Illinois, not much is known. Our lab is working with the state climatologist and other researchers to conduct projects on ticks and climate change in our state. The interaction of climate change, ecology, and tick-borne disease is complex. Increasing temperatures and humidity and extended warm seasons might mean ticks have more and longer days for host seeking and feeding. This would translate into more ticks, more bites, and more disease agent transmission. Increased soil temperatures may mean lowered tick mortality and increased tick activity, again translating ultimately into more disease agent transmission. Climate change effects on tick hosts and habitats could translate into introductions of ticks into new areas and new tick species introductions and increases in pathogen transmission. However, in some areas where tick predators also benefit from climate change or droughts, floods, or fires eliminate tick habitat, we might see a reduction in ticks and consequently tick-borne disease agents. Simply put, we need more surveillance and more active field research to explore these questions. So moving on, landscape management is an essential component of an integrated tick control program. However, the primary driver behind risk of tick bite is human behavior. Therefore, I'm going to present basic information about the interaction between the environment and ticks, information on basic land management principles, links to useful resources on this topic, and then segue to information on how individuals can reduce tick bite risk. Okay, so first, how does a tick find a host? For the ticks of human health concern in Illinois, there are two main strategies: sit and wait, which all species use. They climb up the tip of a blade of grass, wave their forearms around where they've got CO2 detectors embedded in their legs, and also probably radiant heat detectors, and wait for a host to pass where they can grab on. But they also use, if you look on the right at the video there, ambush strategies. Now this is the Lone Star tick, and this is a very common strategy with this aggressive biter. They will pick up CO2 and radiant heat from sources sometimes several meters away. And in this case, it's not even on a blade of grass. This lady was hiding under the leaf litter, picked up on the CO2 from Erica, one of our team members, and came out of the leaf litter looking for her. I've watched Lone Star ticks run across a forest floor for me. It's a bit terrifying when there are a lot of them. So where are ticks found? What does the ideal tick habitat include in Illinois? Well, it's going to be conducive to humid microclimates and tick host presence. Ticks face two major constraints to having a successful life: trying to prevent drying out and finding blood hosts. So you're looking for areas with leaf litter, ground cover and shrubs or understory, canopy cover, and wood debris or rock piles where small rodents proliferate, and slopes where deer like to shelter. This is your ideal habitat, bingo. It doesn't take all these elements, but you are looking for any of them. Now the big exception here is going back to that newly established species, the Gulf Coast tick, which has been described as mesophilic, dry loving. Again, that one you're going to be finding that more often in prairies, fallow fields, roadside ditch grasses. You'll find ticks questing on the tips of grass, sometimes in large clumps, in humid, grassy microclimates, and typically at a higher abundance in the forest and forest edge than on gravel, sand, wood chips, or mowed grass. So you can see in the picture on the right how many ticks Dr. Stafford found when he looked in different environments. And this holds true pretty much across the board except for the Gulf Coast tick. So there are some really excellent resources on landscape management and ticks that I'm going to link to in the next slide. But some very basic tips to prevent ticks is to put a buffer between forests and field or recreational areas. Excellent materials for this buffer are gravel or wood chips. Reduce shady, damp areas where you've got those humid microclimates. Clear brush, rake leaves, and remove leaf litter. Reduce attractions for hosts like plants that they might like to eat, or harborage such as wood piles and rock walls. Mow grass at trail sides. Some of the local forest preserve parks and state parks in Illinois have some really great trailside mowing programs that I think are very effective. If you haven't made your landscape yet, you can design anti-tick landscapes. And finally, put up educational signs so that visitors are educated on tick bite prevention. Again, I think the human component's really big here. So on the right here is a diagram from CDC outlining basic landscape management to reduce ticks. It's part of an outstanding resource produced by Dr. Stafford at the Connecticut Agricultural Experiment Station. The very first link on this slide will direct you to a free PDF of the full manual that he wrote that I consider the tick management handbook, and I encourage you to read through it. The other links provide valuable information on spraying safely for ticks. That Spray Safe, Play Safe project is a really outstanding resource if you're kind of thinking maybe I should be using granules or chemicals at my plot. The other links have information on yard design, educational materials, including signs you can order or print and put up at your site, including our own Illinois Department of Public Health, which is, we have an outstanding vector control team there who are producing new educational materials for free public use. Here are links to some of them, but again, if you're interested in more information, I encourage you to contact them to learn more about what you might be able to share with people who are visiting your site. So I think the primary form of tick bite prevention and tick-borne disease reduction is human behavior. If we can avoid ticks, remove them before they can bite, or remove them properly and quickly after attachment, we can reduce tick-borne diseases. Now, I think when visiting environments that may harbor ticks, a few tricks can greatly reduce tick bites. Because ticks quest on grass stalks or in the leaf litter, they are typically going to transfer onto our bodies at thigh level or below. Then they stereotypically crawl up until they find a place to bite. For that reason, when you're outdoors, regularly scan your body looking for ticks. I start at my torso and then scan down my legs, front and back. I do this every few minutes when I'm walking in the woods. Wear light-colored clothing to make seeing the ticks easier, and make an ascending barrier out of your clothes. Tuck your pants into your socks, tuck your shirt into your pants, and wear a hat and tuck in your hair. Walk in the center of trails and avoid hanging grass. My team also puts double-sided carpet tape around the tops of our boots to stop any ticks from crawling past that point. Now, the second point here, chemical protection, can also be very powerful, but please remember, read labels carefully and use EPA-registered repellents. You can immediately tell, looking at a bottle, if it's EPA registered by looking somewhere on the bottle, usually down in the lower right on the back side, you're gonna see an EPA registration number. Those have been tested. We know that they're safe to use. We know that they're effective. The EPA also has a great repellent choosing tool where you can enter in your personal variables and preferences to find products that are right for your needs. And I link to it on this page. Our team treats our gear with Permethrin, and I do recommend that if you're regularly working outdoors in tick habitats, but remember to read the label. We spray our gear outside, we let it dry thoroughly, and remember that it's highly toxic to cats. So as I'm sure most of y'all are aware, ticks are small, y'all. And when you find one, you'll probably find another soon. Because they are so strongly tied to favorable microclimates, they tend to occur in patches in the environment where these microclimates are. Larvae of some species, like the Lone Star tick, often transfer onto you in clumps. If you have several ticks climbing your clothes, you can quickly remove them with a lint roller or tape. I carry both in my field bag. For leisurely hikes in the woods, I often attach a few strips of duct tape to my pants for quick access. Now, several steps you can take after returning indoors will also reduce tick bites. Check and wash or dry your clothes, remembering that heat, not water, will kill ticks. Check gear and pets. Pet owners are at increased risk of tick bites because pets can bring them inside. Shower within 2 hours of being in tick habitat, and conduct a thorough tick check of your body and your children. Many tick check diagrams are available online. I like this one that I'm sharing from the University of Maine Cooperative Extension Tick Lab, and I encourage you to check out their whole website. It's fantastic with lots of great information. Unlike many diagrams, they make sure to point out to check between your toes. I've heard from a lot of people who do thorough tick checks but miss that part, and then they find an attached tick a few days later. So proper tick removal can also help. Now, I know from personal experience that the first reaction we might have when seeing an attached tick is basically panic. But don't panic. Keep a cool head, follow a couple straightforward steps, and you can remove the tick in the safest way possible. Ticks insert their reverse barbed mouth parts into the skin, kind of like a fish hook. To remove them, you want to grasp the tick as close to the skin as possible. You might pinch your skin at first, and that's okay. You're trying to grab the tick as close to the mouth parts as possible so you don't rip the body off the head. Using firm, steady, gentle pressure, pull straight up. Then wash hands and disinfect the bite site. Once the tick is out, secure in a plastic bag and label with the date, the person, the suspected site of exposure, and store it. I usually tape over the top and place it in my freezer. Contact your doctor or health department for further instructions. So tick removal, what to avoid. There is a lot of bad information out there about how to remove ticks. But in general, what I just showed you on the last slide is the way to remove ticks. You don't burn ticks with a match. You don't smother them in petroleum jelly or nail polish or similar substances. You don't twist the tick. You don't grasp the tick by the tail end of the body or manipulate it with bare hands. None of these are useful responses to attached ticks. And so I want to, in the talk, before we get to questions, to make a plug for our surveillance program and how your organization might be able to participate. If you are interested in having us potentially visit your site to collect ticks or in collecting ticks yourself, please contact me. I'll have my contact information on the next slide and the last slide. I can provide you with instructions to meet those minimum CDC surveillance criteria and with information on how to make a tick drag using easily obtained materials from places like Home Depot or Joann Fabrics, and don't require any sewing and are inexpensive. And then if you can meet our minimum criteria and send us ticks, we can identify and test them and then send you a confidential report. So you'll get a report for your site with the ticks, the identity of ticks, the number, and the pathogens, the disease agents that we found in them. Those results will be put on the IDPH website that's open to the public, but as I mentioned earlier, the results are aggregated at the county level. So your individual site will not be named at that public share. We also have stringent personal protection protocols that we've developed for our team. And if you're interested in learning more about those, because these same personal protection protocols can be great if you're regularly working outside or if you garden tick habitats, contact me. In autumn 2019, we collected ticks all over the state without a single tick bite in our crew. So I'm pretty confident in these protocols. Now, we're going to have another webinar on mosquitoes and mosquito-borne disease agents in Illinois, so please stay tuned for that. I do hope you can join us. We have been learning a whole lot about mosquitoes in Illinois too and have a lot to share. So with that, I'd like to leave this slide of acknowledgements up while I take questions. Conducting statewide surveillance requires a lot of coordination and collaboration. And I'm really grateful for our crew and lab members and students. The vector control specialists at IDPH, Samantha DeBosick and April Holmes, are just outstanding scientists, people, thinkers, and collaborators. All the collaborators and stakeholders who have sent in ticks, and I do want to highlight a great citizen-based resource for the state of Illinois with the Illinois Lyme Association. If you type that into a search engine, you'll get an immediate hit. They are doing great work to advance the cause of tick disease awareness and tick disease reduction in Illinois. And finally, none of this would be possible without the thousands of ticks I have killed. And with that, I would be happy to take any questions you have or clarify any points I made in the talk.
All right, thank you so much, Holly. We do have several questions that have come in through the chat box. If you have other questions, please post them there and I will read them to Holly. So the first comment, one of our listeners has the alpha-gal allergy already, so definitely here.
It's definitely here. Yeah, I'm in a lot of naturalist groups on Facebook where I want to be there if people have questions about ticks, so mushroom hunting groups, or park enthusiast groups. And sometimes I'll post about alpha-gal or tick-borne diseases. And those are usually some of the more popular posts. I'm hearing from a lot of people who are saying they definitely got bit in Illinois, they have gotten a test from an allergist, and they definitely have it.
Wow. The next question is, how did the ticks get here? Do they travel on animals or people or just walk?
Yeah, so typically, ticks are going to be moving over longer distances on birds or large vertebrate hosts. So for instance, with the black-legged tick, the reservoir, for instance, for the Lyme bacterium, the bacterium that causes Lyme disease, the reservoir for that is actually it's small rodents. It's not the big animals, but the reproductive host for the tick. So where the tick is going to be kind of at that final stage where the female is getting a blood meal and developing eggs, those are deer. And the female will usually stay attached for quite a long time. And those deer can travel very, very far distances. So ticks are typically moving around that way, or migrating birds are bringing them in. We think that some of the initial introductions of the Gulf Coast tick, maybe not the current spread, but the initial introductions might have been on birds. And then, I think there might be other factors that merit some more research, such as the effect of flooding on tick movement. Because we know, for instance, that Lone Star ticks, Holly Gaff at Old Dominion University of Virginia has been doing some work where she just kind of said, how long can a tick survive underwater? And so she just kind of dunked the adults down, and we're looking at weeks. They can survive for weeks. So flooding could potentially be moving ticks around as well.
A side note, my son came home when he was really little with a tick. And I tried to run it down. I tried to flush it down the sink, and it crawled right back up. So yes, they could survive in water. That was before I had read enough, and now I know more. Next question.
Yeah, I definitely advise people save the tick.
Yes, and
even if you just put it in the freezer and forget about it, save it, label it, and save it.
Yep. All right. Next question is, many tick management techniques like clearing brush, raking leaves, and removing leaf litter also reduce habitat for other insects that provide food for birds and other animals. How do we reduce the danger of ticks without reducing habitat for beneficial insects?
That is a fantastic question. It's a very nuanced question, and it's also a very important question, not only for the points that were brought up, but for the potential for that habitat itself to be sustaining large arthropods that could be preying on ticks, right? So ticks in their larval stage are preyed upon by larger arthropods, and so you don't want to diminish the population of large arthropods as well. Honestly, I think that's a very nuanced question that requires a much longer follow-up. And one thing that I would definitely recommend is checking out Dr. Stafford's book, the Tick Management Handbook, for more information about that. It's really going to be highly dependent on the site and the particular needs of the site. But I'm always happy if somebody wants to email me to follow up and start a conversation on that.
Great, thank you. All right, our next question is, there are tick bait products out there like tubes filled with materials containing pesticides that you're supposed to put near mice or other rodents, places where they may hang out. Do those work?
It depends. So I just popped up a slide here. That's not as polished as slides that I put in the talk, but I thought that a question about this might come up. It really depends. It depends on the tick density at the site. It depends on the primary tick hosts, especially the reservoir hosts for the disease agents at the site. So for instance, these tick tubes, they have cotton in them that is treated with an acaricide. So that means kind of mite killing or tick killing chemical. And so the rodents can go into the tick tubes. They might pull some cotton out and then put that in their nests. So the idea is that the rodent's essentially seeding its nest with something that will kill ticks on it, and that way you're reducing the tick population in the area. However, some rodents don't respond to those. So it's really going to be very site dependent. Integrated tick control tends to be very dependent on the specific needs of what's going on in that particular site. How dense are the ticks there? What are the species of ticks? What are the species of hosts? How much time can site managers put into it? the tick tubes right now are showing really high efficacy in residential areas in the Northeast, where you have really, really high tick populations, like literally in people's backyards. And that's where they were developed and tested. Now, I think what we're going to see in the coming years is we're going to see a lot more products that can be used in diverse environments, such as rodent bait boxes, where rodents can go and actually eat food, and it delivers an oral vaccine. So the disease agent kind of stops in the rodent. It gets vaccinated against it. But a lot of these are under experimentation or development. And again, can be quite nuanced, might not be cost effective, and are often implemented as part of a broader tick management program. So are often implemented with things like landscape management. And again, if there are any questions about a particular site, please email me.
Related to that, are voles a vector?
Ticks can definitely attach to voles. I've seen
Okay.
on voles.
All right. How long does it take from when a tick bites a person to when it can transmit the disease?
You know, I think there's more research that's continually coming out on this. That question, it's really going to be dependent on what tick species are you talking about, and what disease agent are you talking about, because there are different transmission times for different disease agents. Some ticks take a long time before they even bite once they're on a person. Some ticks, like the Lone Star tick, will bite immediately. And then the feeding behavior of the tick itself varies, depending on how often it salivates, or how long it takes to fully attach, or whether it extrudes a lot of cement. Now, for something like Lyme, the bacterial agent of Lyme, Borrelia burgdorferi, most places I see put it at about 32 hours, although there have been some studies that have indicated it could be faster than that. But it's really going to depend on the tick species that you're talking about, the disease agent you're talking about, and how much research has been done on it. You know, I think that this is kind of a changing target, really. So just kind of keeping up to date on what's going on is the best way. And then CDC is going to have the most highly vetted information.
All right. Thank you so much. Next question is about a Lyme disease vaccine, and the participant asks, many years ago we received just Lyme disease vaccines from our doctor, but since they've been taken off the market. Is there anything being tested for another vaccine? And if so, do you know what the timeline might be?
I don't know the timeline, but I do know, so Limerix is the vaccine that you're talking about. It was, unfortunately, it ended up becoming quite controversial. There is still a vaccine for dogs that you can talk to your vet about. And there is, the last I read, there is a new human vaccine under development. I don't know as much on that end. That resource that I had pointed everybody to, so one, IDPH is a great resource for information on things like this. And then also the Illinois Lyme Association. Jennifer Russell there is extremely knowledgeable on the treatment of tick-borne diseases and the state of current research. She actually serves on the Illinois Lyme Disease Task Force with me, and is just a fount of knowledge about what's going on right now. So I definitely encourage you to contact both of those resources as well.
Great. Next question is about pesticides and how effective are repellents like Picaridin and DEET? Consumer Reports says hours of protection from ticks. Is that true?
You know, that's what you see if you read about these products online. I personally, when I'm out collecting ticks, you know, wearing one of these suits, like I showed you, we embed those suits with Permethrin. I consider Permethrin kind of the front line of defense for me personally against tick bites when I'm out in the environment. There was a study that came out of North Carolina, a very large study with outdoor workers last year, where they kind of followed outdoor workers who were using Permethrin embedded gear and then doing blood draws and looking at any potential toxicity or levels in the blood. And that paper pretty much across the board gave a big thumbs up to permethrin use. I've watched ticks transfer onto my suit when it's freshly impregnated with permethrin and just kind of immediately fall off. It gives ticks, it causes them to have something called hot feet. That's what Becky Eisen at CDC calls it, where they kind of do this little tick dance and they can't maintain contact and they just literally fall off backwards. When I'm hiking recreationally with my family and children, I use both permethrin treated clothing and DEET. I consider DEET to be highly effective, especially because it's kind of doing double duty. It's really super effective against mosquitoes. And so you're getting kind of that double whammy there. But like I said, if you go to that EPA website where you can kind of choose the right repellent for you, and I put the link in the presentation, you're going to be able to kind of fine tune what your preferences and needs are and find something that works for you that we know is an effective and non-toxic repellent.
Great. Two other follow-ups came in on permethrin. One listener asks or mentions that he uses permethrin on horses for horseflies, and it's beginning to show and the horseflies are beginning to show resistance. Is there any evidence that ticks are developing resistance?
I have not seen anything about that.
Okay.
Your observation about horseflies, though, is really, really fascinating to me. Horseflies are kind of one of my personal pet obsessions. And if you want to email me to talk about that more, I would love to hear from you. No, I haven't come across anything or read anything about either a behavioral resistance or a genetic resistance to permethrin in ticks. It doesn't seem like we'd really be using it yet at a high enough level to exert a selection pressure.
That's good to hear. The next permethrin question is, how can one use permethrin and keep one's cats safe? Is it still toxic? Is it still toxic to them when it's dried on clothes or shoes?
No. So the general consensus, what I've read, is that it's when it's wet.
Okay.
So that's a fresh application, but then also bringing permethrin treated wet clothes home. I mean, I haven't seen anything definitive about that, but I've always been quite careful about that. So we have these full body suits that we wear, these full body pocketless Velcro closure coveralls that we wear that are tight at the wrists and tight at the ankles. And when we treat those, you can buy, also I just want to point out, pre-treated permethrin clothing if you don't want to do this. That's cost prohibitive for us. So we have a spray station, a dedicated site that's outside, protected from the wind, where we can hang these suits. So we don't do it on the ground, we can hang the suits, we can spray the suits to achieve the saturation level that it says on the label itself. We let the suits dry for at least 24 hours. And that way we're keeping it away from animals, we're spraying in a safe way. So what I've heard is wet applications of permethrin drift from spraying, and then anecdotally, there could be potential harm from clothes that have been impregnated that get wet and then cats contact them. But again, I'm not a vet. I'm not an expert on this. This is just kind of my personal reading and observations. So if you are concerned, I would really encourage you to contact your vet about it.
Great, thank you. Next question is, aside from possums, what are good predators for ticks?
So I have really heard great things about guinea fowl for the homeowner. I don't know if you've heard guinea fowl. They can be extremely annoying and very loud, but anecdotally, I've heard that they are tick hoovers. That they'll eat a lot of ticks. Now, I heard one person said that it didn't work for them, but pretty much everybody else I've spoken to, it's helped with a home-associated tick problem.
Great. And I'm down to our last question, so if any other listeners have questions, put them in the chat box now. The last question I have is, how short does your grass need to be to make it tick safe?
So what I've seen typically is 3 inches.
Okay. Great.
And I think that if you need a definitive number, then that's what I would put on it.
Wonderful. Nancy has just put up a poll, and I see lots of you responding, but please let us know what you thought of this webinar, how much you learned. And I don't see, oh, I have one more questions. I was bitten and got Lyme disease years ago. Am I immune if I get bitten again?
No.
No.
You know, I actually have, I've seen that information shared before, that if you're bit once that you're immune. But everything that I know about this and everything I have read says no, you can definitely get reinfected with Lyme, even if you've had it before and been treated for it. But again, I'm not a doctor. This is what I have read. These are the things that I'm a vector ecologist. You know, if you want a definitive answer, then definitely reach out to a physician, or again, the Illinois Lyme Association.
All right. Thank you very much, Holly. This was wonderful. And thank you all for participating in today's webinar. We have many more webinars coming up, which you can find on Illinois Extension's local government education website. And we look forward to seeing you all again soon. Thank you so much.
Thank you. Bye, Lisa. Bye, everybody.
Bye.